So thanks, Dennis, for the very kind words. He told me, I guess I was supposed to say, ain't they great? But then he said, I was from California, so I didn't do that. And I had actually hoped that most of you wouldn't come when you'd see that title. You'd be so scared. You'd stay on the beach or golfing or whatever fun thing you do. But instead, here you are, and I hope you enjoy the talk today. Now, we're always technology challenged here. And I'm not even going to try this thing that Clay Cook had such issues with. But I always like to start by pointing out that the work that we do, it's not my work. It's the work of all these people, and I hope I have everybody's pictures up there. There's a whole team of people with different expertise who come together to do the natural products research that we do. And so really, as I'm talking, this is their work, not my work. Let's see if this works. Oh, no, I went backwards. Wrong way. So I'm going to kind of break my talk, into three parts. And the first, I'm going to talk about natural products and what they are. And then I'm going to talk about kind of a theme of there's gold in the freezer, because we have a large freezer full of things, and we have a couple of large grants that we've been working on to pull out compounds from those. And then a little bit at the end, if I have time, on manzamine A, which is a compound that we think has some real interesting properties that might be useful for treatment of pancreatic cancer. Let's see if we can do this. So what is a natural product? So I'm going to kind of read through this a little bit. It's a small molecular weight organic molecule. So if you don't know what that is, don't worry about it, because I'm going to tell you what that is in a minute. They're produced by organisms, so plants or microbes or invertebrates, so lower animals. Some higher animals have them as well, but they're probably made by microbes that live in association with those organisms. They're not essential for the survival of the organism. So they're not like a protein or a lipid or a sugar or all the molecules that you need to keep your body alive. They're something that are made by organisms that maybe we think give them some evolutionary advantage. So if you look at this sponge at the bottom, this is Siphonodyctyon, and it's a boring sponge. It bores through coral heads, and it makes a compound that allow it to colonize space on a reef. And if you look over here, you can see a picture of a beautiful reef in Curacao, one of the prettiest places I've ever been diving. And it's very rich with organisms, and they're all competing for space, and they may have their mate on the next reef track over. And some of these things do things like spawning of sperm and eggs into the water, and how do they find each other? And they use natural products in at least some of them to do that. So those are the kinds of evolutionary advantages. You don't have to have them to live, but they can give you some kind of advantage. So I'm going to define some chemical jargon. We've had a few people come through the lab recently, and we throw out terms. You know, I'm an organic chemist. That's what my degree is in, and I've spent a lot of years learning organic chemistry. And I always assume everybody in the room is an organic chemist and that you know exactly what I mean. But you might not know what I mean. So I'm going to try to define what these are. You know, what am I talking about when I use those terms? So what is a molecule? And it was interesting because I went out to the internet, which is, you know, the source of all knowledge now, and I did a search, and I found these definitions of molecule that really confused me, and I thought, man, I don't know, I'm a chemist and I don't know what that means. But this one wasn't too bad. It's the simplest unit of a chemical compound. You like how they define it as a compound? That can exist consisting of two or more atoms held together by chemical bonds. So a chemical compound, on the other hand, and this was from a really neat website called Chemicool, is a substance formed by the reaction of two or more chemical elements. So, you know, down at the bottom you can see water, which is hydrogen and oxygen. Those are both elements, and they're in a molecule. And the next part is that the elements in a compound are present in fixed ratio. So water is always H2O. It's always H2O. It can't be anything else. That's the stable structure for water. There's another definition that we had people visiting the lab, and they were a little confused because pharmacists sometimes make compounds. They take different organic or different molecules, and they put them back together, And they make something that's like a pharmaceutical compound. It's a drug that you would use, and it's made up of more than one thing. And so when I kept saying, well, we're working on a chemical compound, they kept saying, but you just purified it. Why are you mixing it back together? You know, what's wrong with you? So for those of you, what I really mean is something like water. It's got more than one element. They're put together with chemical bonds, and they're in a set ratio. That's the kind of molecule. Now, what's an organic molecule? So we all know about organic farming, and everybody wants to have organic food, and it's better for you, and you know, all these things. But when you're an organic chemist, that's not what you're talking about. You're talking about molecules that are made up of carbon and hydrogen, and sometimes nitrogen, oxygen, sulfur, or phosphorus. But the basic rule is if you're an organic molecule, you have the element of carbon and hydrogen in your structure somewhere. And so I have all of those circled here. Now, our natural products from the sea can actually also have fluorine, chlorine, bromine, and sometimes iodide, which are all halogens, and they come from seawater and they get incorporated into the structures. And many molecules that are just important in life processes and keeping you alive also have things like sodium, magnesium, potassium. Some of the metals are in them as well. Part of the enzymes, they have those as well. But for us, organic molecule has carbon and hydrogen, at least. So chemists use these little complicated things, and we always throw these up at you and expect that you know exactly what these structures mean, and you're able to recite them back to me later on. But I just wanted to walk through kind of it's kind of a shorthand, like somebody who can write really fast, they just do a shorthand. This is the molecule sucrose, so that's table sugar. And it has, wherever there's a little elbow or an intersection of lines and it doesn't have an oxygen or something else, that's a carbon atom. And on that carbon atom, there are rules for how many things carbon has to be attached to. And so chemists know that up here there would be 2 hydrogen atoms there, and one here, and one here, and one here. And so we actually can then put this together and come up with a chemical structure. And so we know that that molecule sucrose is something that in space, all the atoms, so the hydrogens are white and the carbons are black and the oxygens are orange, and we know where they are in space. And it's really important for us to understand that if we're organic chemists, so that we can know how to make them and make them better and things like that. This is Splenda, so sucralose, and it looks much like sucrose, except for there's some chlorine atoms that have been stuck in, and this sugar is actually slightly different in terms of the way the oxygens are facing. And then I just thought I'd show a natural product down here that's in the new sweetener called Truvia, which comes from the plant stevia, and there's actually a whole series of what are called stevioside or steviol compounds, where this is steviol in the middle, and they have different sugars. And you can't actually break this down in your gut, so that's why you get the sweet flavor, but you don't actually get any calories. And it's been approved and can be used now. So that's a chemical shorthand. So when I put those up, you'll now know that everything like that translates to something like this. And some of them translate to even more complicated. I had to bring this. This is a structure that Priscilla Winder in our laboratory has been working on for quite a few years. And I think she's finally there and it's done, and she's got the paper written up. And it's a really complicated molecule. It took a lot of work to get that done. It's very impressive. So why do we want to work on natural products? I mean, what's the purpose? Well, it turns out that 62% of small molecule agents, or small molecule, little ones that are not large molecular weights, that have been approved for use as drugs can be traced back to natural products. So they're either the natural products themselves, or they're a simple analog of the natural product where we've changed the structure ever so slightly, or sometimes synthetic chemists, so that's chemists who make the compounds in the laboratory artificially, might use our compounds as kind of the vision or the idea behind making a better compound, one that lasts better in the body or that has less side effects. But they use ours as the lead compound around doing it. And so aspirin, you can actually chew on the willow tree or birch tree bark, and you'll get a compound that converts to be exactly the same as what aspirin converts to. And you'll get those same effects. Morphine, I think people all know, comes from the poppy. Penicillin, we've all heard of the penicillin drugs, comes from a fungus. The statin drugs, the very first of the statins, came from a fungus, and those are important for cholesterol lowering. And then many cancer drugs, so Adriamycin and Taxol, are used all the time, and those come from either a bacteria or a yew tree. And on the next slide, I'm going to show you a couple of the marine natural products that have been approved for use, and there's more and more coming along. This one's only been approved in Europe. It's called Yondelis or Trabectedin, and it comes from a little tiny tunicate that you can actually go see up by the inlet. It grows up there, and it also is very common throughout the Florida Keys. And in fact, the samples that were collected to do the structure actually came from those areas and also in the Bahamas. This compound is called halivin, and it's actually where they used a molecule found in nature, and they found they only needed half the structure. There's actually another big group, I think it comes off over here, but this was the part that was the really functional part of the molecule. And it's been approved for the treatment of breast cancer, but only after the person has failed many other therapies. Now in Germany, they've actually found that this compound is so good, they've moved it to be the first-line therapy for a number of people. So I think it's really going to take off as a great compound. The one at the bottom is actually interesting because it's a different approach to cancer treatment. So this part is what we call a warhead for an antibody-directed conjugate. And so this is a molecule that's very closely related to a marine natural product called Lovastatin 10. And Lovastatin 10 was the inspiration for this, which is called monomethyl orstatin E. I don't know where that comes from, but it comes from, Lovastatin came from a little mollusk. This is a spacer and this is a linker, and then it's tied to a monoclonal antibody, which is a molecule that can then recognize the surface of other molecules on the surface of cancer cells and direct this drug directly to the cancer cells. So it shouldn't affect many other cells. It should really go to the cancer cells. And so it's kind of, they've always looked for the magic bullet, and this one seems to be doing okay. And it's kind of interesting to me because with a different monoclonal antibody than the one used in breast cancer, they're actually targeting pancreatic cancer, and their phase 1 results are back, and they actually saw a response in the patients. So it's an exciting new approach, and we hope some of our compounds end up being on the end of one of these warheads someday. So the central dogma of all natural products is where you have biological diversity, so different organisms, and it can be, this is sponge and corals. These are microbes that we've isolated, Peter McCarthy's group. You get different chemicals. And hopefully what you can see is that these all just look different. So you can imagine that the models for those are going to look different. Those are all different structures, and they may all do different things. And we have a very large freezer that I think it's probably about 4 of them would probably fit in this room. So it's a pretty big freezer. It's filled with these bins, and in the bins are a whole bunch of different organisms. And so we have this very large collection of organisms that we can work on to get natural products from. We also have a really large collection. This is Peter McCarthy, and he has about 19,000 microbes that he's isolated. These are just examples that come from inside the various organisms, the sponges and the corals, or even from sediments. And so it's a very large collection. And once again, it has the potential to have lots of different compounds present. Now, we have this big collection, and we can start to test it, but the world has changed a little from when I came here in 1928, not 1928. Okay, I came here before my mom was born. 28 years ago. It feels like 1928 today. Okay, I woke you up. Anyways, the world has changed. We used to just test crude extracts and go through a big, long bioassay process, and anyways. But now there are laboratories, and this is one up at Sanford Burnham. It's one of our colleagues' labs, and they have these huge robots. They fill whole rooms. I mean, they probably fit 4 of these robot systems in this room. And they can test 1 million samples, so 1 million different things in a 16-hour workday. So that's a lot of things. And so if we want to work with groups like this, we have to change how we work. And so the other thing I wanted to point out was that these are what are called chromatograms, and each one of these little peaks here is actually a different compound. And so we know that we have interesting compounds present. And so what we've done, and what's been funding a lot of research in our laboratory, is received 2 fairly substantial grants that have allowed us to tap into the diversity in that freezer and analyze the extracts and determine which ones might have really interesting chemicals and start to fractionate those out. And then by having them more pure when they go to our colleagues, we can work with them and much more rapidly be able to identify active compounds. And we have two different grants, one we call the Grand Opportunity and one called the Molecular Roadmap, which is a big NIH initiative. And so I want to just walk through how we're making this library of natural products, and I'm going to do some little demos here, and we'll see if they work. I don't know. You never know if they're going to. But I want to start with what's the first step. So the first step is we make an extract. So does anybody drink coffee? Anybody? Raise your hand if you drink coffee. Okay, tea? Anybody drink tea? Okay. So when you take your coffee beans and you grind them up and you put hot water in there, you extract out chemicals, natural products that are present in the coffee beans or in the tea. And one of those is caffeine. I mean, I'm sure many of you like caffeine. There's its little structure, and it comes from the coffee beans. There's also lots of flavor components that are present in the coffee that are also natural products. The way we extract stuff is a couple of different ways. One way is we have a little grinder, and we just grind it up, and it's a little organism daiquiri. You know, put a little sponge in there, put some ethanol in, and you grind it up. Just don't drink it. And that makes an extract. But we also have this. It's really an ugly machine. I went and took a picture of it. It's really ugly. It's gray. But it works under high pressure and temperature. And we put the sample in a little tube, and we put high temperature and pressure solvent through, and we use a tiny amount of solvent and get a really good extract. I don't know what the equivalent for coffee would be, maybe espresso or something where they use these really higher temperature water. But that's actually part of our process here at Harbor Branch to cut down on the amount of organic solvents and our waste product and our waste stream, and to be a little more green is to use that machine because it uses a lot less. So we extract using that. And then this slide is to remind me to tell you that, and to kind of show you that there's a lot of different compounds. Each one of these little dots, these are all different extracts of a sponge called Batzella. And I guess they decided I'm not doing the demo yet. Thanks, Jill. So all these little dots are different compounds. And so if we want to find out which one's active, once again, we have to separate them and get them all pure. So that's kind of the goal of the process we're doing. So the method that we use to purify things is called chromatography. And it's really called the type we use most often is adsorption chromatography. And there's a general rule that likes attract likes or likes dissolve likes. And we all know that things that are similar, they get together and they like each other. And so let's see what my next slide is here. Oh, I want to tell you about polarity of molecules. So up here I have water. And you probably know this, but in case you don't, each bond has 2 electrons making up that bond. And so you can see the electrons in this bond, and then there's another water. And here on the bottom, I have a substance called heptane, which is a hydrocarbon. So you can see it's all carbons and hydrogens. And oxygen is what we call electronegative. So that means that it wants to grab all the electrons and pull them to itself. That's what it likes to do. It's really kind of piggy, but when it does that, let's see if it works. It pulls them closer to itself and further away from the hydrogen atom. And we can see that again on the other one. That's one of oxygen's properties. When it does that, we end up with kind of a charge. So there's more electron density, and they're negatively charged over here. And there's more just the hydrogens without the electrons around them over here, so those are protons. So you end up with what's called a dipole. And so water is considered a polar molecule. So because it has this property that the oxygen is kind of a little piggy and pulls the electrons towards itself. On the bottom, we have the heptane. And carbon likes to share. I don't know how to describe it exactly, but it doesn't want to pull anything towards itself. It really wants to share its electrons. And so these are equally shared, and you have no dipole across that. So what we say about heptane is that it's a nonpolar molecule. Okay, so let's see. So here, Jill, is where I'm going to actually try to see if I can make something work. So, and for those in the back, I hope you have good eyes. I don't know how this is going to work. I haven't actually done it here. Now, for those in the front who we pass glasses out to, if you don't wear glasses, put them on now. You're in the splash zone. Okay, you're not really. If I hit you, that'll be really bad. The one thing I just want to show you is that I actually, maybe you can see that there's a line there. It's like the lava lamp or something. And I can actually, I always like to vent it. Let's see if I can vent it properly so it doesn't build up pressure. You can shake it, and it'll go to 2 phases. So what I have on the top is heptane, and what I have on the bottom is water. So nonpolar substances don't particularly like polar substances, and they separate out, okay? So that's something, and think of like the oil spill. We had all the gasoline, or not gasoline, but you had hydrocarbons floating on the surface of the seawater, and they didn't mix very well. So now the tricky part here. 1, can I get that off? And 2, let's see. I have this weird mixer that I'm hoping I can get some of both phases. Let's see. Okay, without spilling it all. So I'm going to try to add this in here and hope I get some of both. I'm just squirting in some substance. And then I have to find this. And what you might be able to see is that there appears to be something orange there and something blue there. And if I shake it up, I'll get green or something like that. Let's shake it up. Oh, is it going to work? Yay, it worked. What I have here are 2 substances. So if you imagine that this was actually an extract of a sponge, and I put it in where I have heptane and I have water, the things that are nonpolar will go into the top phase because that's what they're soluble in. And the things that are really polar will go into the bottom because they're like water and they want to be in the water. And there are always compounds that are kind of half in between. So half of them are going to go in here and half are in here, so you have to do multiple of these shaking around things. But in case nobody believed me, I'll just shake it again, and then look what happens. They always separate out. So that principle is one way that if I have an extract of a sponge with all those different things in it, and I just add it to a couple of solvents, I can partition it and separate things right away. I can open the stopcock and put one in one flask and one in another. But it's one way of separating things. Now let's go to the next, I have to go do the next slide. Let's see if I can do that. So just so you know what I have in here, I actually have beta-carotene, which comes from carrots and things like that. And on the bottom is FD&C blue number 1. And I'll just point out that if you look at beta-carotene, it's all carbons and hydrogens. It's a hydrocarbon. That's why it happily wants to go into that top phase. And the FD&C blue number 1 has all these sulfates, which are charged parts of the molecule, and that makes them very polar like water. And that's why even though there's lots of carbons and hydrogens in here, it wants to go into the water. So that's the kind of principle of this partitioning that we do. But the next step is to actually use other methods. And so we usually use column chromatography. And I actually brought in a whole bunch of different columns. So you can see these are all HPLC columns. And this column is for our combi flash system. And what they all have in it is some kind of a solid. So there's a solid in there. And rather than using 2 liquids, I can use a solid that acts as though it's has polarity like water. And then I can pump a solvent through that's like heptane, or is heptane. And then compounds will actually move along the column and slowly start to separate. So let's see if I can, I've lost this. I just want to point at, this is silica gel down here, and these are these OH groups, so it looks a lot like water, so that's very polar. And the solvent, we would pump it down the column, and we'll actually see the compounds move based upon their polarity. They'll just sit there and kind of keep partitioning between the surface and the liquid that we put through. And this is actually Priscilla working on an HPLC, which is the kind of machine that uses one of these big guys or this one. And before I spill it, I want to cover that back up. So that makes sense to anybody? Or are you all lost? Okay, not totally lost. Okay, so my chemists spend quite a bit of time developing separation systems to separate compounds that are very similar to each other. And it's really kind of an art, and they're all amazing at doing it. Now we have a little video here that, let's see. This is kind of the time lapse of where there's a solid thin layer plate, And then there's a liquid moving up it. So I think you can see the liquid moving up it. And all the compounds are partitioning and moving up, and you can actually see all these different colored compounds. If you can imagine, that same kind of thing happens inside this column. It's just not on a thin a plate, but it's actually inside the column. So I don't know if that was easy to see or not, but that's just another example of chromatography. So we use, for our grants, our major tool is this thing called a combi flash, and it's the one that uses this size column. It allows us to work on pretty large scale and to separate things out into a lot of different fractions. And this is an example of what we'll get. And so across the top, these are HPLC chromatograms where we're analyzing, and wherever you see a little blip like that, that's a different compound, one or more coming off. And when we do the work with this, the chromatography with this column, we end up with fractions where this fraction is almost just this one thing. And this one is these things, and this one is these things. And if you can imagine that if I send, well, there's two things that happen. This one's now highly enriched. And if we had sent this extract to a lab that was looking for something that that doesn't kill cancer cells, but that changes a protein in the cell, and in the extract was one fraction that just kills every cell it sees, and another fraction right here that actually does that response that we want, we would never be able to detect this compound. But by testing this fraction, we can see that because this compound's not present. And so that's really the advantage of making this kind of library of natural products, is we can start to see these trace compounds and what they actually do. So we have the library. What are the kinds of things that we're doing with the library? And I'm just going to give you 3 examples. So the first one has to do with a grant that we have. We actually have 2 grants to look at anti-malarial agents. And you may or may not know that there are 216 million cases, clinical cases of malaria every year. And that 665,000 people die every year from malaria. And 86% of those deaths are children. And so it's really a pretty devastating disease. Turns out there are also 3.3 billion people who live in places where malaria is endemic. And not only that, but we send our service people overseas to places where malaria is, and some people go on vacation where malaria is, and you want to be able to have a treatment if you should get the disease. The latest treatment is based upon a natural product called artemisinin, and even that particular compound has developed resistance already in Thailand. And it's probably because it was used there traditionally. The plant that gives it is called wormwood. So there is a need for new drugs. There's also resistant for the therapies. And although in the United States it's not currently a problem, and the reason we don't have to worry about malaria is that we control the vector, which is a mosquito. But there are cases every year that the CDC will report where someone goes overseas and they get infected, and then a mosquito bites them, and then it manages to spread it around, and you'll get these little pockets of malaria. So hopefully nobody here ever goes to one of those places and brings that back to us. But it certainly can happen. So we have 2 grants, one to work on marine invertebrates, and another one that we just received that Peter McCarthy is the PI on for marine microbes and finding compounds from those. We're working with Devapam Chakrabarty, who's up at University of Central Florida. He's at the Burnett School, medical school there. And he's one of the people who worked on the genome of the malaria parasite, which is called Plasmodium. And we tested just a really small part of the library, just, I don't know, a few hundred fractions. And from that, we found 8 actives. Now, I have to tell you, when we selected the ones we sent, we were pretty careful to select things we thought might show some interesting properties. We applied for a grant, tested more, and got even more actives. And so, just as an example for how fast we can get things, we had this sponge, Spongiosauриdes, and we sent these fractions that were enriched. We came back and did one analysis that told us the molecular weight of the compound present in that fraction, and it turned out to be nortopsentin A, and it was the active compound. And the next slide kind of shows what happens when you treat cells with nortopsentin A. So here's the cells, and they've been infected with the parasite, and this is just treated with solvent control. And these little green, see all this purple in there? That's what happens over 48 hours to the parasite. So it just keeps going through various life stages, and at the end it looks really very bad to me. And I'm not an expert in malaria, but that doesn't look good to me. If you treat with nortopsin A, on the other hand, it just freezes it at a stage that's called the trophozoite stage, and then it doesn't go further. And we're still doing more work to see whether this would be a real potential therapeutic, and that's going on in Debapom's lab. So that's one example. So another example, and I'm not going to show you any structures here yet because it's something that's just happening in the lab, and maybe 3 years from now we'll have some, well, sooner than that we'll have compounds. Maybe I'll talk in 3 years about it. But tuberculosis is another disease that I didn't realize how many people die from it, but 1.5 million deaths per year from tuberculosis. And some people may think back, and that was a disease called consumption that I thought had kind of gone away, but apparently there are still many people dying from it. What's interesting, too, is that there are 2 billion people who are unknowing carriers of what we call latent TB. So it's a bacterium that causes it, and it sits in the macrophages, and it doesn't really cause any effects. But if you become immune suppressed or something like that, it can cause an infection that then you have to be treated. And the current drug treatments for TB are a cocktail of drugs, and it's administered for 6 to 9 months. Yeah, that sounds like fun. And they said that many people just stop taking the treatment. They get tired of it. It must not be pleasant. I don't know. And so drug resistance is becoming an issue. And it's also a big issue in the population of people who have HIV because it synergizes with HIV and makes HIV much, much worse. So one final fact about TB is that there have been no drugs that work through what we call novel mechanisms. So every new drug that's been brought on to treat TB works the same way as every other drug that's been around, and since 1963. So it's been quite a long time since somebody found something new. So once we had the library, we were contacted by a fellow who just joined, once again, University of Central Florida, and it's Kyle Rohde. He's a new assistant professor there and an expert on TB. And he said, you know, I'd really like to test your library against TB. And so he has assays that are very different than what people have traditionally used to try to find new drugs to treat TB. He has ones that look at this latent form, and also that are only, you can detect things that the bacterium, I'm not really good at this, the bacterium infects macrophages, and then you can actually treat with some of our compounds, and they're not active against the bacterium alone, but only in the macrophages. So there's some kind of stress process going by, and it makes it really unique and different that we've actually found hits. So he has tested a small subset, and we found 6 leads, of which 2 of them hit in this really unusual model, and so you wouldn't have found them through any other way. One of them, we kind of know what the compound is, but we submitted a grant, and we just received notice that it's going to be funded. So we get to follow up on these 6 as well as work on screening the rest of the library. So that's a new thing in our laboratory to work on TB. So the next little example has to do with pancreatic cancer, and I think some of you may know from talks in the past that it's the 4th leading cause of cancer deaths. There's less than a 25% survival rate 1 year post-diagnosis and less than 6% 5 years post-diagnosis. So it's still a very difficult one to treat, and it's a very aggressive form of cancer, and you'll hear a little bit more about that when I talk about manzamine A. Esther Guzman has been working on a protein called nuclear factor kappa B, and all I'm going to say about it here is that it can be responsible for driving some forms of pancreatic cancer, and that kind of making it aggressive and being able to block this particular protein might be a good way to intervene in the disease. And so Esther did some screening with a grant that she had and found a sponge called Amphibleptula madrepora as being a potent inhibitor of NF-kappa B. So she screened this subset of the library, and we were rapidly able to do the chemistry. Here's the sponge. And the active compound is this unusual peptide called microsclerodermin A. And what was interesting to us was that we had actually found this compound before, and we actually have a patent for it for anti-tumor use, but we really didn't know anything about how it worked. And so right now in Esther's lab, they're working a lot more to figure out whether this compound's really going to be useful and how it's really working and through this nuclear factor kappa B. So that just shows we can pretty rapidly get to compounds, and that's 3 examples. So I don't know what time it is. Oh, good. I have a few more minutes. So what does the future hold for the library? Well, we're actually screening it broadly. That was part of what we told the NIH we would do when they gave us money to make it. So we are looking at neurodegenerative disease with a researcher at University of Florida. He's at the Brain Center up there at the med school. We're working with Moffitt Cancer Center. They're sending just a subset through 9 different anti-cancer assays they have. We have a company that's testing some of the materials. We have already tested it for compounds that will block the spread of prostate cancer to the bone and found some hits there. We've been looking at a new pathogen, well, it's not new, but one that people are looking for new compounds to treat, Clostridium difficile. And then we have a lot of assays here at Harbor Branch, and there's so many that I'm not even going to try to list them. And then I wrote others not yet conceived of, because people call us all the time. They hear about this library. They see how fast we can get to interesting structures which might have potential. And so they contact us fairly regularly. And my theory down there is make the library, and they will come. And in fact, it has worked. That's what we put in the grant proposal, and it has in fact worked. So now I'm going to change gears and talk a little bit about this somewhat complicated story on manzamine A. And I'm going to try to see if I can make it not too confusing. Let's see. This is the work of Esther Guzman and Georgios Kalafatidis. Did I say that right? Yeah, Esther's saying yes. And he is a postdoc here over the last few years. And, well, anyways, I'll just start to tell you the story. So manzamine A was originally found way, way back in the sea farm. Remember they said I came here as a postdoc with the Sea Farm Project 28 years ago in 1928. Yeah. Anyways, they found this compound, and it was a fellow named Tatsuo Higa, who is a truly wonderful natural products chemist. He was working here as part of the Sea Farm Project. He had his students here. He's from Japan, the University of the Ryukyus. And they found this interesting compound called manzamine A. And they knew that it had some anti-tumor properties. They went into animal models, and they saw activity. Since that time, a lot of other groups have found activity. So they've shown it has anti-malarial activity, anti-neuroinflammatory activity, anti-HSV is herpes simplex virus, so it hits a virus as well. So that's kind of a broad spectrum of activity. But Esther was working on, when she was a postdoc here, I think, or right after that, I don't know, was working on manzamine A and running an assay and found that manzamine A had the ability to do, so these are pancreatic cancer cells, and if you look at how they grow, they're long and thin, and that's how they grow. And they're very able to spread. They don't have a lot of what we call cell-cell connectivity, and they're not touching each other. And normal cells do. They grow right up next to each other, and they touch each other. There was a control drug, because we thought we knew how it worked, that we were using, and you can see it now makes these much more flat, and they start to touch each other. And HB071 is manzamine A. It's our little shorthand name for that. And you can see how it also converts them from looking like this to looking like this and this, which is much more normal. So that was exciting. That was one of the good things about manzamine A. Another thing about manzamine A was that Esther was able to show that it blocks cells from migrating. So all these little dots, there's an assay where you can watch the cancer cells move towards a kind of a nutrient gradient, or you can put a membrane in, and wherever all these little dots are, those are cells that have migrated. And this is just solvent, so that's kind of a control. And this is this drug that we were trying, and then here's manzamine A. And it's really hard to see many dots down here or here. There are a few. But most of the cells don't migrate. So that's exciting because you might have a compound then that can block the metastasis or the spread of the tumor, which is one of the things you want to do in pancreatic cancer. And this really busy slide, I'm not going to walk you through it. All I want you to get is the message here is that manzamine A can sensitize cancer cells to what's called programmed cell death or apoptosis. So manzamine A alone is not very cytotoxic, but it can make it so that drugs work again. Drugs that don't work will now start to work, and they'll induce apoptosis in the cells. So that was another hallmark of manzamine A. So then we had the opportunity, working with Novartis, which was a partner on a national cooperative drug discovery group, to send manzamine A to them and say, they had this really cool thing called the hip hop, and I was going to have a little music play here, but I decided not to. It was developed at Stanford. I don't know where they got that name, but well, I do know, but I won't go into it. It has to do with haploid insufficiency profiling and stuff like that. But here's the way it works. yeast has 2 copies of each gene. So just, I don't know if that makes much sense to people, but it has 2 copies. And if we knock out one of them, so it's not there, it's not able to do anything, and if a gene is important, we then treat with our compounds all these different yeasts that have all these different mutations where just one gene, one copy is knocked out. If they have that happen and that gene is important to how your drug works, you'll get reduced number of growth of the yeast that have that mutation. And you can measure that using some really cool bioinformatics that if anyone wants to know, I'll try to describe it to them later. The HOP assay, you knock out both copies, and if the gene isn't important, cells grow normally, or the yeast grows normally. And if the gene is important, you get changes in the number of the cells once again because they're trying to compensate in some way. And so you get this really complicated data set. And I'm just going to show these diagrams. Don't spend much time on them. But here's what happens. They use a computer and a big program, and they say, what is the profile of our new compound, we don't know what it does, look like compared to all the compounds we do know how they work? And you look for matches. And so what they came up with was they said manzamine A is an uncoupler, like a compound called prodigiocin, that affects bacular ATPase function. So the answer of all this fancy genetics they did was that it maybe is targeting bacular ATPase. And so now you're all going, okay, yeah, so what's that mean? Thanks so much, that's great to know. Anyways, so why do we care about bacular ATPase, and especially in pancreatic cancer? So what is a bacular ATPase? And some of you maybe came to Clay's talk last week, and you remember these little hydrogen atoms, or these protons, and that's acid, so think of acid molecules, not LSD, but acid like HCl or something. I have to stand close to this because I have to make this work. The bacular ATPase is this little thing I have drawn here, and this yellow ball is going to be kind of it's something called a lysosome or a vacuole in the cell. And the bacular ATPases are supposed to take the hydrogen atoms and or the acid, and pump it into these cells and make them highly acidic. And then under those conditions, inside this little organelle, things that your body doesn't need anymore get degraded and turned back into something your body can use for something it needs, because there's always proteins you don't need anymore and other things, and so it just degrades them. So this is like a little garbage disposal of the cell, and it needs acid to chew up the molecules. So the bacular ATPase pumps the protons in. So there you go. That's the role. It just pumps them in. Now, what do cancer cells do? Well, they stick these things out on the cell membrane. And then what happens is they start pumping them out of the cell. And so you can actually just see these things getting pumped out there. And so in effect, around the tumor, there's this little pool, it's not really a pool of acid, I'm exaggerating, but it becomes very acidic on the outside of the tumor. And that leads to a lot of really bad things. That leads to resistance to drugs. So the drugs that should work don't work because now you have this acid pool out there and it's affecting the pH of the drugs. It leads to the spread of the tumor. So the tumor is now driven to move to another place. It facilitates the movement of the tumor or the tumor cells. And it can also drive the just proliferation, getting those cells to grow and divide and just become worse. So this acidic tumor microenvironment that's caused by the bacular ATPases being in the membrane where they shouldn't be. In normal cells, they're not there. They're in here, and they're doing what they should be doing. And in here, they're on the outside. They're pumping this little acid pool on the outside. I always want to say, wasn't there a James Bond movie where there was the acid, you fall in the acid pool and you get dissolved? It's something like that. Anyways, so if we treat them with a bacular ATPase uncoupler, or proton pump inhibitor, that's another word for it. And so we put a little block on there. Then what happens is these don't get very acidic, but also this acidity doesn't get pumped to the outside. And then there's this process called apoptosis or programmed cell death that you want to have happen. It starts to happen again. The tumor is inhibited, and you can also have drugs that didn't work before start to work. So that's all something that we want to have happen. So it turns out that these compounds that can block the function of this bacular ATPase may be very useful in treating certain forms of cancer where this kind of thing is going on. So some of the data that George then did to show, okay, we got this answer from this hip hop, is it real, is it not, was he took the cancer cells, and this is just treated with solvent. And there's always a little acid because there's a process going on in cancer cells whereby they're always putting out a little acid. But you can see this is a known proton pump inhibitor or vascular ATPase inhibitor. They're very green. That's the acid. Actually, it's using a dye that can sense the pH in the cell. So they're very acidic, stuck inside. It's not getting out anymore. It's getting stuck inside. And so there's manzamine A. Looks much like this other control drug. So that showed that we think we're on the right track. And it is affecting vascular ATPases. So now there's an even worse concept than the vascular ATPase. There's something called autophagy. And autophagy can be a driver of pancreatic cancer. And I'll see if I can walk you through that. So I have here this complicated thing. It's a process of lysosomal cell digestion to provide nutrients. So it's where those little organelles I told you that were filled with acid start, they go through a whole process, and I'll show you a little picture of that. But they start eating up things and providing nutrients to the tumor. And I'll see. Let me go to the next one, I think. But it's something that happens in many pancreatic tumors, and it can be induced by chemotherapeutics. And here's this little complicated diagram. But here's that little, what was yellow in the last one, where the acid is in there. It's called the lysosome. And it fuses with something called an autophagosome. And you get this fused form. And then you get another thing called the autolysosome, I think. And then that starts just degrading all kinds of proteins in the cell and providing energy and building blocks to the cell. And typically, autophagy happens when there's cell starvation and the cell needs nutrients for itself and it's not getting enough nutrients. It will start to kind of take its own spare things and convert it into energy and nutrients and building blocks. And there's a protein called LC32 that the levels of it increase at this point, and then eventually it gets degraded itself as the process goes on. And so you can use it as a marker to see if you have autophagy going on in your system. And I'm just going to show one of these diagrams because I know it's kind of complicated. But this dot here is the manzamine A, well, first non-treated and vehicle control. There's not very much of this LC3 protein. So the darker the dot, the more of this protein there is. And here in manzamine A, in 2 different tumor cell lines, that's ASPC1 and PANC1, we're getting these large increases. And George went on to show that, in fact, it never gets degraded. So what we're actually doing is blocking autophagy. And so that's one of the things manzamine A does. So why do we care? So here's the pancreatic cancer cell. And it turns out many of them themselves are not doing this autophagy. They're not eating up their own cells. What they actually do is they send signals out, and they start causing all the cells around them to undergo this process. And as those cells are kind of broken down, they feed the tumor. And so the tumor is kind of cannibalizing all the tissues around it. And that allows it to grow and to get out of control. And so if we can block the autophagy at this stage, then we may be able to block the feeding of the tumor. And manzamine A seems to do this kind of process. And there's actually a compound that is a malaria drug that has very similar properties. And it's actually in clinical trials right now for this exact reason, because it can block the autophagy. It's one of the hydroxychloroquines. And so it may be a new treatment for pancreatic cancer. And manzamine A may also be the same. So I'm sure that was very confusing. So, but we think that there's some real potential there. So there's lots of people, their names are a little tiny because there's so many of them who are working hard on all these projects. And there's been a lot of funding from the National Institutes of Health and multiple different, NCI and NIAID and General Medicine and NCAM, which is National Complementary Alternative Medicine, which funded the library generation, and then NOAA for some of our collections. And so with that, I think I'm done. And